Thank you for the previous presenters for this nice introduction. Makes my life easier, so I will be able to skip a few details. So indeed, I am a business developer at Serendip Innovations. At Serendip Innovations, we are an innovative biotech company with core competency in the field of vectorization, pretty much the same as the other companies. We have developed a novel and non-viral vector to transport biological assets. And as it's very novel and to gain adoption by the biotech pharma world, we are currently developing a drug candidate in the form of cancer vaccine to obtain proof of concept in the clinics. So today I will not talk about this asset. I will mostly focus on our vectorization technology. So if we summarize it to the core, there are two approaches. Either you want to deliver a protein or a peptide. Do I have the... Yeah, a protein or a peptide inside a cell, or you want to deliver a gene inside a cell that will ultimately produce a protein or a peptide in vivo. So there are numerous kind of applications that you can do, and I will go over them a little later in the presentation. So what do you actually need from a performance delivery system? You want it to be targeted, allowing intracellular delivery, for example, and other advantages. In number two, you want it to have optimal pharmacological properties, avoiding liver, having good exposure, having low immunogenicity of the capsid itself, and so allowing repeated dosing and so on. In three, you want it to be easy to assemble, and this is particularly of interest when dealing with drug discovery and lead optimization steps. And in four, you want it to be easy to manufacture for obvious reasons. So you see me coming with the non-viral vector that we have developed. We checked all the boxes or will check all the boxes. And yeah, so please keep in mind the 4 different points, but because I will get back to them in just 3 slides. So what does it do first? So it's a programmable vector. So it's a vector for which you can simultaneously program the inner and outer surfaces with different things to match different applications. For example, on the left there, you can actually produce a cytotoxic peptide on the inner surface and deliver this capsid with an antibody fragment expressed on the outer surface to target cancer cells. But you can also transport mRNA coding for a CAR, for example, and deliver it to a T cell to transform it into a CAR T cell in vivo. You can transport peptides or mRNA vaccines or antigens, and deliver them to dendritic cells to trigger an immune response. But you can also do more fancy things. You can actually have a hybrid expression of capsid monomers expressing different antibody fragments and turn your non-viral vector into a T cell engager, for example. And you can actually think of many other applications, but not described today. So what is it exactly? So it's a plant-derived virus-like particle. It's 30 nanometers in diameter, has no lipid on its surface, and it's actually coming from a grapevine virus originally. So as I just mentioned, it's programmable simultaneously on the outside, on the inner and outer surfaces, and it can transport RNA, peptides, and proteins. It is produced in plants. It is a very original way of producing vectors. We understand that, and we think it is actually a very good advantage, as it's already compliant to GACP and GMP standards. I will come back to it a little later. So now I'm coming back to my 4 points. So these are key differentiating advantages. The first one is the targeting delivery. So in this example, we customized the VLP to produce on the inner surface an antigen, and on the outer surface an antibody fragment targeting dendritic cells. So first, we showed that it's actually binding via ELISA test, very simple testing. Then we moved in vitro, in cells, so targeting those. So the target cells are actually in green there. They are expressing the antigen, and the virus-like particles are in the vicinity of these cells, so you can actually really technically see how well they target those cells. And then here we sorted the cells so we sorted the target cells, those that do not express the target from those that do express the target, and showed that if there is some targeting of the VLP, there is some binding, very efficient. And if there is no DC targeting, there is no binding. So it's very clear proof. And then we moved on to an in vivo design. It's an immunization design of mice in which only 100 picomoles of the antigen in the form of a peptide allowed to trigger an immune response in mice. So if we move to the graph on the lower section, we can actually see that only the VLPs that simultaneously contain the antigen in the form of a peptide and are directed to the dendritic cells are able to produce a very efficient specific lysis in vivo. So we believe this was achieved thanks to the fact that this very low amount of antigens were actually densified within the VLP, resulted in an improved potency and a better therapeutic index. So I'm moving on to other pharmacological properties. This is the body distribution. So this is the naked, so we use the naked VLP, so no targeting on the outer surface, and the inner surface is coated with a reporter nanoluciferase. And then when we administered that to mice, dissected them, and looked at the fluorescence in the organs. So on the upper section, we see the quantification of that and actually see that there is a very broad distribution, quite even, with low amount to the liver and main retention in pancreas, bladder, GI tract, kidney, and some lymph nodes as well, which can be also very interesting for some applications. And so in this one, we showed that there is no pre-existing anti-VLP immunity in human. We used the serum of 13 human healthy donors and showed that there is, technically no previous anti-VLP immunity. This in red is the positive control. And then we moved on in vivo. So this is a very complex design. I will not go over it today because it would be way too long. But what we wanted to show here is that there is no impact of the immunogenicity of the body against the VLP itself on its repeated efficacy, dosing after dosing. So in this one, especially the green group, we repeated the dosing and that allowed it to maintain an antigen efficacy. Here, and lastly, in the pharmacological properties, we showed that it has a very safe profile in terms of safety biomarkers and also on the toxicity on human PBMCs as well. So now I'm moving on to my third key differentiating advantage. So it's the ease of assembly. First, it's fast to screen. So you can actually test different drug variations or modalities of your drug asset in the form of a DNA sequence that you actually insert into a plasmid here. These are then transferred and transformed into planta. I'm not sure if you're not familiar with plant transformation, it actually goes via an agrobacterium and then in plants. It's very robust, easy, and straightforward transformation. And The moiety here, this is a monomer expressing capsid monomer, the therapeutic moiety in the form of an mRNA, peptide, or protein, and a cell targeting moiety on the outer surface is all expressed in a single step in Planta. And then there is an auto-assembly of 60 nanometers also in Planta, and it's an integrated design, and you obtain this in just a single step. The leaves, you can actually freeze them and store them for a long time. There is a purification process that is very similar to the one of viral vectors, and in the output you get a very consistent and robust drug product that you can actually store for a very long time at 5 degrees. And here I'm talking about the VLP in itself. And then my fourth key differentiating advantage is the ease of manufacturing. Very obvious, as you can just spread out the cultures to obtain more products. Guidelines are already here. Others have done it before. It's all compliant with GACP and GMP practices. As I mentioned, there are already 3 examples of products following that process on the market already. And also you have very favorable end product features in terms of stability, purity, particle size distribution, and also reduced costs. We were able to produce up to 2 grams of VLP, which is huge, in a single R&D batch. And we have an estimation of less than €1 per milligram of the final drug product. And last but not least, it's not written here, but there are no known plant viruses that are harmful to humans. This is all protected by intellectual property, by patents, obviously. We're continuing to file new patents to protect modifications and improvements of the technology. And our IP strategy also relies on the secret on the know-how, especially concerning the production in Planta. Just a short word of our first drug candidate in the form of a cancer vaccine targeting the indication MSS colorectal cancer, for which current standard of care is unfortunately not efficacious. We're doing this in collaboration with a hospital and academic groups in France. There is actually one asset currently in clinical trials relying on a cancer vaccine that is delivered with an LNP. And our objective is to obtain the same efficacy than this competition, but with lower and repeatable doses, triggering lower inflammation, more flexibility in terms of drug regimen, and being also more affordable. We already obtained major advances at preclinical stage with cell targeting confirmed, redosing possible, and no liver distribution confirmed. So this is our founding team made of 3 people. And thank you very much for your attention. Here I leave you with the contact address if you want to contact us. Thank you very much for your attention.
Merci beaucoup, Jean-François. Donc, de la même manière que pour les deux précédentes présentations, si vous avez des questions, ne pas hésiter à les adresser. Je pense qu'il y en a une, Jean-François, pour vous. VLP looks good, but are the antibody fragment on the surface of VLP to guide and the delivery payload inside is attached by any linker? Moi, si vous voulez la relire pour être...
Well, we use different kinds of antibody fragments. They actually do work. And it's actually a very good question how to attach the payload inside. If it's a peptide, you can actually, well, there are different methods actually. You can actually customize your DNA sequence right from the beginning to include the peptide on the N-terminal side of your DNA fragment so that it's expressed, or there are different ways to attach it to them, but this is kind of a secret for the moment. And in terms of RNA, it's kind of the same. It's kind of there is an attachment mode that we keep secret for the moment. I don't know if I answered the question correctly.
Il y a un thanks, donc j'imagine que oui.
Okay.
Any other question?
Yeah, it has to release the payload at the end. I mean, it depends on the application. I mean, if you need to release the payload, obviously there can be some attachment mode that needs to be released inside the cell. For other applications, you might not need to release it. Actually, it can be still attached to the VLP, and as long as it's being exposed at one point, can also work in some circumstances. Need to be tested. Thank you. Thank you for the great work.
Perfect. Anyone else? Quick question to Christine and Jean-François. Jean-François peut-être et Christine après. Have you demonstrated lower? Go ahead.
Yes, thanks for the question. Yeah, that's exactly what I tried to show in this complex design experiment, this one, in which we repeat the dosing and show that there is a repeated efficacy, and it's so there must be some. I mean, yeah, there is some. We tested actually the immunogenicity against the VLP. It's growing. There is one because it's obviously you administer something, so there is a growing immunogenicity, but this immunogenicity is not sufficient to hamper its efficacy.
Jean.
Christine,
Christine,
maybe you want
peut-être?
to.
Yeah, we have done that as well with multiple dosing of the particles, and we have been able to see increased antibody responses, but antibody responses directed against the antigen of interest, but not against the particles themselves, and no inflammatory responses so far.
Merci Christine.
Thanks.
Quelqu'un a une autre question aussi pour Pascal? Là, il nous reste 5 minutes pour Pascal ou AReNA. N'hésitez pas. You can ask any question to our three speakers. Feel free. As we said, so the webinar is recorded. So if you have any question, you can also address us some question. I'm going to share our email address. So feel free to send us any question to AReNA, and so we will put you in contact with the three speakers if you want. So that's something we can do. Ok, je pense qu'on a fait le tour des questions. Donc, écoutez, pour clôturer ce premier webinaire, je tiens à remercier nos trois présentateurs, donc Pascal, Christine et Jean-François. Je tiens à préciser que ce n'était pas intentionnel d'avoir trois entreprises françaises aujourd'hui. C'est un hasard qui nous a amené à ça, effectivement. Mais en tout cas, effectivement, je souhaitais vous remercier et puis d'avoir fait l'effort de présenter en anglais pour notre communauté bilingue de Montréal. Donc, merci à vous trois. Et
Merci à
puis,
vous.
écoutez, j'espère qu'on aura des opportunités d'avoir des collaborations entre la France et le Québec au travers de vos trois présentations.
Merci
Avec
beaucoup.
plaisir.
Merci.
À bientôt.
Merci.
Merci à tous et puis
Bonne
bonne journée
journée,
à
au revoir.
tous. Merci.